You know, when I've reached out to the other four to see if they would charge me to ship there. That's my budget. Twice the amount. I have got a microphone too.
Oh.
That's the question.
Oh, okay. Yeah. Okay.
Official.
Okay, good.
So I'll just enter the question to Jeff.
Yeah.
What question I'm sure will ask you.
Yeah. 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 Okay.
There's 3 participants already.
Good.
I mean online.
There are 2 participants here.
Yeah.
Hi.
My name is Lee.
Yes.
That's good. Well, sort of.
So, yeah, in some respects it's very good. But we have kids coming up. And it will. We'll do that. But prior to that, we did a prep. And it worked. And the work, I'm just saying, came to the observations, okay? Right. That's it. No, I know he's very mean. Okay. He was a senior mean. All right.
How about you when that's you?
Yeah.
Well, he does go last, so we'll okay. As long as we get it by like, because we don't do this. Yeah. I would also kind of sense tonight because all this no. Oh, gotcha. Yeah, you're right. Yeah. Yeah. Oh, congrats there. Wow.
We did send it to admin before.
Yeah. Yeah.
What happens when you leave an open print stage? Huh? Well, it's your email.
Info. Yeah. Yeah, maybe. I guess. Yeah. There is not much traffic, so maybe we can. Hello, Sandra. Good. How are you?
I'm well.
What happened?
Years ago, something wrong with my elbow.
Oh no, sorry about that.
Trying to get the information down because that's the first thing they're going to ask me when I go in. Have you been noticing?
No,
And it's
yeah.
in such a weird spot.
Oh, sorry.
You got your microphone's hot right now.
Yeah.
Okay, I just want to make sure because you're in conversation with everyone right now.
All right. Maybe, yeah, if you want to.
Is your microphone's up?
It's on. It's on. I turn it off.
Okay. Good morning. Thank you so much for joining us today. Just want to introduce quickly this seminar. So we are sponsored by 10x Genomics, Illumina, and the Genomic Core here to all came together to provide this technical seminar. We'll have two researchers' presentation as well as a 10x and an Illumina presentation. So thank you so much again, everyone, for joining.
Hi. Good morning, everyone. My name is Egon Anghini. I'm the Science and Technology Advisor for 10x Genomics. And well, first of all, thanks for joining us here this morning. So I'm going to give you an overview of what we provide. And of course, if you have questions, just feel free to reach out to us. If you would like to have this presentation, absolutely, we can send you the PDF after the seminar. So don't worry about like taking screenshot or pictures. You can have those slides. So a couple of words about 10x here. So we are a young company, but very innovative. And over the past 7 to 8 years or so, we released more than 20 unique solutions in the single cell field and now also in the spatial transcriptomics arenas. And more products are going to be available very soon. And actually, I have some slides toward the end of the presentation that shows you what is coming up. Essentially at 10x, we have 3 main pillars. The Chromium platform is what includes all solutions for single-cell genomics. Then in 2019, toward the end of 2019, I would say, we released our Visium platform, which is for spatial transcriptomics. And now we are currently developing our 3rd pillar, which is a in situ analysis solution. So this is called Xenium, and we'll use a new instrument which will allow you to interrogate gene expression with subcellular resolution. And it's an in situ type of analysis. And so instead of interrogating the whole transcriptome, you'll be looking at hundreds of targets and proteins also if you want to. And I have a couple of slides at the end of the presentation. Now, regarding the single-cell platform, what I'd like to say is we are really proud to see that the Chromium solutions, the Chromium platform has been included in the list of top 10 innovation by the scientists multiple times now, and also featured in top-tier journals. And more than that, we are always super excited to witness the type of research that is being made possible thanks to 10x. And so now we have more than 3,200 publications using this technology. And so first of all, what I like to say is, so why do we do a why do we push a single cell approach instead of bulk type of analysis when we interrogate a sample? That is because each sample is really complex and so formed by many different types of cells. And so if you were to use a bulk type of analysis, what you'll be doing or will be reading is an average signal of, let's say, a gene across all cell types. And so let's say you do bulk RNA sequencing, you'll be measuring an average signal of, let's say, CD45 across all cells that are present in your samples. And so this is not really a method that allows you to get resolution or throughput. And so with the single-cell genomics approach, you essentially interrogate each individual cell in your population, and that allows you to identify the different subpopulations, identify rare cells, and even find new cell types. And here what I'm showing you is a typical t-SNE plot of gene expression data where each dot represents an individual cell, and cells are clustered together based on their gene expression profiling. So cells with a similar expression profiling will cluster together because they will belong to the same subpopulation. Now at 10x we have what we call a end-to-end workflow, where essentially all reagents that are required to make a library are included in the kit we provide. And so all you need to do is to, when you run 10x, is to prepare a high-quality single cell or single nuclei suspension. And then on the other end of the workflow, we also provide a fully integrated bioinformatics pipeline for data analysis. And those come at no cost for you, so you just need to download them from our website. And, the video works, good. So here essentially you can see how the technology works. And the idea is to partition individual cells or individual nuclei and barcoded gel beads into those little droplets that we call gel beads in emulsions or gems. And inside those droplets, the cells are lysed and their contents, so in this case, let's say the RNA is released and captured by the oligos that are attached to the gel beads. And so just for now, we're going to focus on the first type of oligos, which terminates with a poly-DT tail. And so this is the oligo that will capture the poly-A mRNAs. And from this point on, what we do is to generate a full-length cDNA, which will be barcoded. And then we're going to use a little bit of the cDNA to make a sequenceable library. Now, there is also a 10x barcode attached to each gel bead, and this sequence is unique to individual gel beads. So different gel beads will have a different 10x barcode sequence, and this is what essentially gives us the single-cell information of the data, because by reading that barcode, we can tell what are the transcripts that are coming from the same cell. And the UMI here stands for unique molecular identifier. This is a unique sequence that allows us to count the copy of a particular transcript present in a cell. Now, just give you an example of how this technology was used in the field. And here we have a science paper published in 2020 For this particular study, the authors wanted to look at the tumor-infiltrating lymphocyte in two cohorts of melanoma patients. That one cohort that they call complete responders. So there's essentially where patients that could still respond to the adoptive cell therapy, and non-responders that they did not respond to the therapy. And so they used the single-cell gene expression solution to analyze approximately 20,000 cells across those across 10 patients, 5 and 5 for each cohort. And they found that they essentially identified 9 main clusters of cell population. And the complete responders cluster here in this subcluster A, the non-responders cluster here in the subcluster B. So there was already a nice separation. And because of that, the authors wanted to look a little bit more in details into those clusters. And when they did that, they found two completely different T-cell subpopulation. And so the complete responders, there are those one here, they presented a subpopulation of T cells that were negative for CD39 and CD69. And those were, essentially they showed a more stem-like phenotype. And this phenotype was associated with a good disease regression and a good TIL persistence. Whereas the non-responders, that those are here in black, they did not have this stem-like population. The T cells here were more differentiated, and those T cells essentially were associated, or this phenotype was associated with poor TIL persistence. And so you can appreciate that this type of data is made possible thanks to a single cell approach, because thanks to that you can actually identify different subpopulation of cells, and this also will be important to guide therapies, because at that point it will be a better choice to try to infuse CD39 negative T cells that might give you a better tumor control over the other population. Now, measuring the RNA sequencing or measuring gene expression is great, but it's not the whole story. And that's because biology is very complex. And what we want to do here is to try to capture multiple types of information simultaneously from the same cell. And this is made possible thanks to what we call feature barcode technology, which essentially is a way to measure gene expression plus something else. And this is made possible thanks to the other class of or types of oligos that are attached to the gel beads. And so the idea here is to, let's say, measure gene expression plus some other type of information. Oftentimes, this other type of information is in the form of the expression level of cell surface protein. And so how do you do that? Essentially, you start with your single cell suspension, and you label your single cell suspension with oligo-conjugated antibodies. Those oligos attached to the antibodies will have a capture sequence which is complementary to the capture sequence we have on the gel beads. And so when you generate libraries, at this point you're going to generate at least two types of libraries. That will be your gene expression libraries and your cell surface protein libraries. And when you do your data analysis, you'll be able to simultaneously measure gene expression and cell surface protein expression from the same cell. And taking advantage, this is essentially what people call site-seq. We call it feature barcode, but the technology is essentially the same. And by taking advantage of feature barcode, you can measure different types of analytes. So cell surface protein is just one. But then you can, for instance, use oligo-conjugated dextramers that carry specific antigen to measure the antigen T-cell receptor binding. Or you can even measure CRISPR-mediated gene expression perturbation using this feature barcode technology. Now, the 3' or single cell gene expression is only one solution we have. The other gene expression solution we have is a 5' chemistry, and is what we call the immune profiling solution. And this is a great tool to study immunology, because you can simultaneously measure gene expression and profile the full-length paired VDJ sequences of the T-cell receptor or the B cell receptor, or both. And again, this information comes from the same cell. And if you now take advantage of feature barcode, now you can really measure up to 4 different types of analytes from the same cell in just one assay. And that will be your gene expression here. You can profile the TCR or full-length TCR or full-length BCR or both. If you label cells with oligoconjugated antibodies, you can also measure cell surface protein. And if you have T cell, you can measure the antigen binding specificity with those T cells. And so just an example of how this technology was, this particular kit was used in the field. Here we have a COVID paper. And this was published in Nature in April 2021, so almost 12 months ago. And in here, the authors profiled 700,000 cells from 2 cohorts of patients. And what they were trying to answer with this paper, or the question we were trying to answer was, why do kids typically show a milder disease when it comes to COVID compared to adults? And so they isolated PBMCs and cells from the upper airway tract and trachea and bronchi from pediatric patients and adult patients, and control as well. And then they generated gene expression libraries, TCR and BCR libraries, and also site-seq libraries. So those are feature barcode libraries. I can't remember exactly the number of proteins they screened, but it was in the dozens. And so, and by doing that, so they generated a true multi-omic data set. And by doing that, they were able to demonstrate that, well, actually, pediatric or kids, they showed a stronger local production of interferon compared to the adults. And the adaptive immune system of kids was more, showed more a, I would say, how can I say, naive. That was what I was looking for. A naive phenotype and a way more and a higher chronotype diversity compared to adult. And that actually seems to help kids fighting COVID better than the adults. And again, it's a great example of why you want to use a single cell approach, and the type of information you can gain when you actually combine gene expression with this something else that can be protein expression or cell surface or VDJ clonotyping. Now, switching gear a little bit, still a multi-omic solution I'd like to talk about, but here is it's a kit that allows you to measure gene expression and chromatin accessibility from the same cell. It's what we call the multi-omic kit. So why do you want to do that? Well, it's because we know that there is this unique interplay between epigenetic regulations and gene expression. And so if we really want to understand how genes are expressed or cell lineage is established, we really need to capture both types of information simultaneously. And in all fairness, you could do that even without the multi-omic kit, but the method would not be, that strategy would be far from ideal, let's put it this way. That's because you would start from one sample, but then you will need to split the sample into 2 aliquots, make 2 separate libraries, and then use a bioinformatics approach to essentially infer linkages between gene expression and chromatin accessibility. Now, oftentimes you're going to essentially have cells that will be misaligned. Rare cell will be very difficult to identify. And generally speaking, every time you have a region of open chromatins, those methods will assume that you have a positive regulation of gene expression, which is not the case. And so we come up with a new solution. Now, what you can do is to start with a single nucleus suspension. Those nuclei will be transposed in bulk and then partitions. At the very end of the workflow, you still generate 2 separate libraries. But those libraries will be directly linked together, because if you look at the gel beads here, now we have 2 different types of oligos. One oligo will still capture the mRNA. The other oligo will capture the transposed DNA fragments. But critically, those 2 oligos will share the same 10x barcode. So by using the information of the barcodes, we can now directly link the cells in the ATAC space with cells in the gene expression space. What is really important to see here is that pretty much every time you analyze a sample, even a simple or a sample like a PBMC from an healthy individual, every time you add ATAC on top of gene expression, you'll be able to better separate cell population. Like here we see thanks to the ATAC, we were able to identify a cluster of naive B cells separated from a cluster of memory B cells. Whereas if we were to rely on gene expression only, well, we will just see one cluster of B cells because gene expression in this case is similar between those two subpopulations, but the chromatin accessibility, the chromatin landscape is significantly different. Now, because you also have the two data sets linked together, what you can do, and this is a screenshot from our Loop browser, so this is the analysis you can do very easily at your laptop or your computer, is to identify putative regulatory elements that are linked to a particular gene of interest. And so, for instance, here we have the CD56 gene, so NCAM1 is what codifies for CD56. And here you have region of open chromatins identified by those peaks across all different subpopulations of cells that we annotated. And as you can see here, this region of open chromatin is positively correlated with this gene because the expression of CD56 is very high here in just the CD56 positive cells and not in the other cells. But on the other hand, here we have another region of open chromatins in myeloid monocyte cells. There is a red arc. That means there is a negative correlation between open chromatin and gene expression. And when we look at the expression level of these genes in those cells in particular, well, the expression is pretty much zero, is extremely, extremely low. So again, an example of how you can use the two, or can take advantage of the two data sets to really start understanding the biology that lies underneath your sample and gather critical information that otherwise would have been missed just by measuring gene expression or ATAC only. And of course, the question is now, I generate all this data, how do I analyze it? Well, so what I showed you here is a screenshot from Loop, and Loop is our desktop app that allows you to interact with the data and really explore the data set. It's a desktop app, so it runs on Mac or Windows. We have fantastic tools in the form of our FAS or tutorials that will help you get started with the analysis. And on top of that, you can run our pipelines using our clouds for free. So this is a 10x cloud. You'll be uploading FASTQ file on the cloud. And this will allow you to run the pipeline, the Linux-based pipeline for free and without the need to have a Linux dedicated machine. Now, what the two papers I showed you before, so the first one profiled 20,000 cells, the second one 700,000 cells. Granted, the second one was a large study, but it shows you a trend. And that is a trend that we have seen occurring in the field over the past couple of years or so, where investigators tend to analyze more and more cells. That is because the more cells you analyze, the more data you have, and a better understanding of biology you can get. The problem is that, as you may imagine, analyzing a lot of cells, it might be very expensive, and the protocols to scale up sample prep oftentimes are cumbersome and not very easy to implement. And so because of those challenges, we wanted to come up with a new solution, which is in the form of a new instrument and a new assay. So the new instrument is called ChromiumX, and the new assay is called the high throughput assay. And when you now combine the ChromiumX with the high throughput assays, now you have a platform that allows you to run hundreds of thousands to millions of cells with a significant reduction in cost per cell. Now, just want to clarify one thing. So the ChromiumX is the instrument that we consider to be essentially the future for single-cell genomics. That's because, true, it's compatible with the high throughput assay, but it's compatible with everything we have available right now. It is also compatible with future applications, and I have an example in a couple of minutes. And then it's the only instrument that is compatible with the high throughput assay that come in a 3' or 5' chemistry. But keep in mind, the ChromiumX can run everything. And when I say high throughput, again, here is what I'd like to clarify that, typically when we talk about high throughput, we immediately think about, oh, I want to profile a large number of samples. Well, that's definitely a possibility or an option. To me, that would be the least common option. What is going to be way, way more common is to profile more cells from pretty much the same number of samples. And this is actually what the high throughput assay allows you to do. That's because there is a new chip architecture, or microfluidic architecture, I should say, compared to the standard. And thanks to that, now what you can do with the high throughput assay is to capture up to 20,000 cells per sample, as opposed to the 10,000 cells per sample that you'll be able to capture with the standard. The capture rate is still the same, but again, crucially, the multiplet rate now for the high throughput assay is half of the multiplet rate that we have on the standard. So it's 0.4% every 1,000 cells compared to 0.8%. So that means that, if you were to run an entire chip, an entire high throughput chip, you have 16 wells now instead of 8. And if you take advantage of the maximum number of cells per sample, you will be able to profile up to 320,000 cells per run. And that can be 1 sample, 2 samples, 4 samples, you can decide. And this option does not include cell multiplexing or cell hashing. I don't have the slide here, but if you take advantage of cell hashing or cell multiplexing, that 20,000 actually goes up to 60,000. So what you can do when you profile way more cells per sample is actually to identify rare cells. And so here we have a lung tumor samples that was profiled with the high throughput assay and the standard assay. We were able to identify pretty much the same subpopulation of cells, but because of the higher capacity of the high throughput assay, we were able also to identify those rare tumor cells that we essentially missed with the standard assay. That's not because the chemistry is less sensitive. The sensitivity is exactly the same, but that simply is a question of numbers. So more cells you capture, the better is your chance to identify those rare cells or identify new cell types. Now, that is a kind of scientific justification. Now, financial justification could be, well, now I have to assay. I need to decide when it makes sense from a dollar standpoint to use a high throughput assay. And actually, again, this is a very, is a kind of 30,000 feet view, and we can go into the numbers in much more details. But essentially, there are 2 conditions, 2 scenarios that you need to meet in order to start saving money on the high throughput assay compared to the standard. The scenario number 1 is actually shown here in the graph. You need to run at least 2 samples and capture more than 10,000 cells per sample. In that case, if you use the largest kit size, you will be saving more than 50% in 10x reagents. The second scenario is to run at least 4 samples on a high throughput chip and capture any number of cells. So this could be 20,000, 10,000, 5,000, 3,000, you choose. So as you can see, it's, meeting those 2 scenarios is not hard at all, and it will actually cover probably most of the experiment. Those 2 scenarios do not require any cell multiplexing, so there is no cell hashing and anything. If you do take advantage of cellplex, there are some situations where, or cell multiplexing, there are some situations where the saving will be even higher. And again, this is a very quick overview. We can go into the details with you in much more details. So now we talked about gene expression. So there is one of the new solutions we're going to have available soon, and soon means mid-2022, so still a few months, is a fixed RNA profiling solution. Now, this kit will be compatible only with the Chromium X series, so it's not going to run on the Chromium controller. And it's one of those examples when I was showing you the slide with the showed future application compatible with the Chromium X. Well, this is the very first example. It's a brand new kit, and it's going to be compatible only with the Chromium X. That's because the Chromium X is really the instrument for the future. And so what you can do with this is to collect samples as they become available, fix them in 4% PFA, and then essentially block or preserve the biological state of the samples and run them at a later time point. So it uses a brand new chip, a brand new chemistry. And for the chemistry, we use a probe-based assay where set of pairs of probes are tiled across transcript from approximately 18,000 genes from humans or mouse. And so the workflow here is, again, end-to-end workflow. What you'll be doing is to fix samples in PFA, then add the probe set to the samples before partitioning cells under the chip. After partitioning, after generating gems, the probes will be barcoded, and then they will be sequenced on an Illumina sequencer. Now, you can see that when we look at different storage lengths, so here is 1 week or 1 month, a 4-degree 1 week or -80 1 month. For PBMC, we didn't see any differences in the relative proportion of the cell subpopulation. So that is regardless of the length of time that those samples were stored. And then, of course, we want to compare the fixed RNA profiling with the gene expression, so with the traditional 3' gene expression solution. Again, we found a very high correlation when it comes to gene expression sensitivity between the two assays. Here we combined the fixed with the fresh data set. And again, when we look at the genes that were expressed in these samples, as you can see, those genes are expressed in the same cluster for both assays. I didn't mention here, but feature barcode is also compatible with the fixed, so you can still do protein expression and gene expression from the same cell. The other advantage that is made possible thanks to the probe approach, probe chemistry, is that now you can multiplex samples without the need for oligoconjugated antibodies or oligoconjugated lipid molecules. And that's because we attached a unique barcode to each set of probes. And that allows you to essentially collect samples as they become available, and then batch them the way that makes most sense for your experiment. So you don't need to, first of all, buy antibodies, label cells when you label the samples as you start the experiment. So think about how you want to do this experiment, but simply the labeling is essentially already included in the chemistry, so you can multiplex them the way that makes the most sense. And in the last 5 minutes or so, let me show you also what is coming soon. So from the single cells side, we're going to have a nuclear isolation kit that will be compatible with a very wide range of frozen tissue. And all you need is essentially an hour of your time and a benchtop centrifuge. So here the sample will be lysed or homogenized, dissociated in a lysis buffer, then pass it through a column. Then you need to wash nuclei, count them, resuspend them, or resuspend them, count them, and load them on our assay. They will be compatible with our single nuclear gene expression, or the ATAC assay, or the combined multi-omics, so gene expression plus ATAC. And again, this kit will be available mid-2022. So there's going to be a solution that takes advantage of the feature barcode technology to profile antibody-antigen interaction or antigen T-cell receptor interaction across tens of thousands of cells. This is what we call BEAM, that stands for barcode-enabled antigen mapping. If you are familiar with LibraSeq, this is essentially our version of LibraSeq, with the differences that is fully supported, including the bioinformatic pipeline, and is also compatible with T cells. And so, just to give you an idea, we screened more than 4 million B cells that were incubated with the spike protein, for spike proteins from the SARS-CoV-2 virus. We found 55 human broadly neutralizing antibodies and two pan-coronavirus antibodies. That is in a span of less than a week. There is also going to be a new assay. So this is a 5' CRISPR assay. So far, if you are interested in CRISPR perturbation, the assay we have available right now is compatible with the 3' chemistry only. with this new assay, we also going to make CRISPR compatible with the 5' immune profiling solution, so that, again, from the same cell, you'll be able to capture gene expression, the cell surface protein expression, and of course, the guide RNA where they are, and interrogate how genes are expressed, and of course, measure TCR and BCR from the same cell. And from the spatial transcriptomics side, we're going to have a new slide that will allow you now to use oligoconjugated antibodies. So this is essentially site-seq, but for spatial transcriptomics. And so thanks to this new slide, you'll be able to profile whole transcriptome and profile doses of proteins, or the expression level of doses of proteins simultaneously from the very same tissue sections. Again, available pretty much, they say available mid-2022. A new instrument called CytAssist, which will allow you to generate Visium libraries starting from pre-mounted or pre-archived tissue sections. This essentially will allow you to, so you'll place a Visium slide in a CytAssist, and then the instrument will permeabilize and transfer RNA or RNA and proteins from the pre-mounted slide onto the Visium gene expression slide for downstream analysis. And a higher resolution version for Visium. So here, each barcoded spot will go down, will be smaller than 10 micron in size, which will allow you to get single cell resolution from a tissue that you are interrogating. So that will be great for developing tissues such as this eye in this mouse embryo, or very heterogeneous tissue, can be pretty much any form of cancer. And the last slide, so this is Xenium. So it's our new in situ platform for in situ analysis. It's a new instrument, will be compatible with fresh frozen and FFPE tissue sections, will allow you to interrogate the expression level of hundreds of genes with true subcellular resolution. You'll also be able to profile the proteins together with RNA sequencing from the same tissue sections. It doesn't use an NGS readout, so this is a microscopy-based readout, and imaging and data analysis are integrated in the instrument. And yeah, that's it. So that's your local team. So I'd like to introduce my colleague Yves-Marie Walter. She's your sales executive. Heather Eckhart is your sales associate. She's not here. But she probably will be watching the seminar right now. You have two field application scientists, Cara and Joseph. Joseph is what we call tissue specialist, so it's essentially field application scientist for everything related to spatial transcriptomics. And then you have two science and technology advisors. One is myself, and Leilani. She is the spatial science and technology advisor, so we'll be focused more on our spatial transcriptomics and in situ platform. And with that, yeah, thanks for your time, your attention, your time, and I'll be happy to take any question. I'm not sure if we take question now or later on. Do you have time? Okay. Yeah.
So first question, is there a size limitation for cells that can be loaded on the Chromium X or the controller?
Yeah, it's a good question. So yeah, there is a size limitation. So what we tested in-house and what we officially support are cells up to 30 micron in diameters. But having said that, we have many customers running larger cells, let's say between 30 and 50 microns successfully. Now, if your cells are significantly larger than 30, and so larger, let's say if they start becoming like 55, 60, and above, that might create a challenge because the limitation is really the size of the microfluidic chip. So if that is the case, what we typically recommend is to prepare nuclei. That will solve the problem on the side.
Thank you. The next question, is there any cell type that cannot be loaded or cannot be used on the 10X instrument?
No, good question. No. You can profile pretty much anything. As long as they go through the chip, yeah.
Perfect. And then question for people that were attending virtually. Can we do cellplex with single-cell nuclei for RNA-seq?
Oh, good question. So no, yes, I'm sorry. Yes, cellplex is compatible. Now, if you are interested, and that is 3' is single cell 3' only with cellplex. So keep in mind that. So if you have nuclei and you want to run, for instance, the multi-omi kit, which is gene expression plus ATAC, cellplex is not supported and is not going to work because the multi-omi kit, the multi-omi gel beads don't have the capture sequence for cellplex. But yeah, if you have nuclei, it's compatible for 3'. Perhaps just reach out. It's always good to have a conversation when it comes to cellplex, and we just want to check the quality of samples and so. And our FAS can definitely help.
Just for clarification, the platform is not capable of handling prokaryotic cells, correct?
Yes, yeah, I'm sorry, yes, that's a great point. I was thinking in terms of eukaryotic cells, mammalian cells actually. So that is a great point. Now there is a preprint where investigators from Europe, I believe it's Belgium, they profiled E. coli and I think Bacillus. But anyway, they were bacterial cells. So they had to come up with a completely different strategy for, and so they designed probes that will target bacterial genes. And those probes will have a poly A tail. They have a bioarchive manuscript. I haven't seen the peer-reviewed manuscript yet. I think it's still in bioarchive, but this is to say it's possible. It will require a significant degree of optimization. But yeah, you're right. So that the technology has been developed to essentially capture poly-A mRNA, and so that is mammalian cell and yeast. That is sometimes oftentimes we get question about yeast. Is that possible? It's tricky because you need to you essentially need to remove the cell wall. I'm not aware of any publication with yeast, but again, it's one of those things that if you are interested, you can maybe think about strategy.
Thank you so much.
Perfect. I'll introduce the next speaker.
Can everyone hear me? A little bit higher. Sounds good. Perfect. Hello, so thank you all for being here this morning. My name is Logan Silber, and as mentioned, I'm the sequencing sales specialist at Illumina. I actually graduated here from the University of Wisconsin-Madison in 2015, so very excited to be up on this stage since I've seen so many presentations here before. After graduating from the university, I moved to the University of Minnesota Genomics Center, where I worked there for the past 6 years, helping a variety of PIs and postdocs with their experimental needs. And during the end of that period, I really started to help out a lot more with their 10x applications as well as their spatial applications in that sense. So with that, I'm just going to go through the agenda very quickly here. So we've heard a good deal about single-cell sequencing and sort of the applications for that, so I'll give a recap of that. From there, we're going to go into the two instruments in the Illumina portfolio that are best suited mostly for single-cell sequencing. We'll be discussing their technical advancements that have made them the right choice for this application here. And then from there, we'll give a few examples of 10x experiments on an Illumina instrument and how best to determine your experiment parameters in that case. So with that, So I'm sure everyone here has probably heard this analogy before between a smoothie and fruit, which for bulk sequencing and single cell. So when you're performing bulk RNA sequencing, you're measuring the average expression across a sample to identify differences between cell types there. So you aren't getting a specific read on the amount of the gene expression for each cell type specifically, you're just getting the average there. So when it comes to single cell analysis, hence the fruit, you're able to pick apart and figure out the gene expression for each specific library. And just to show a little bit more about what single cell sequencing can be used for, it can be used to assess cell-to-cell heterogeneity, as shown previously. Researchers who have used single cell sequencing have been able to find very rare cell types in tissue. And I've seen papers out there using single cell sequencing to help map cells' trajectories. For example, for brain development and neurological development, cell types can go through many different types of pathways, and so by utilizing single cell sequencing, that could be a possible application for using that as a experiment. You could dissect transcriptional mechanics, and you can infer gene regulatory networks here. So I just want to showcase these possible applications that you can gather from single cell sequencing as some of you here perhaps start to work on your grant and think about how you would like to, what you would like to focus on as you move forward here. So with that, there are a lot of different single cell sequencing applications. We have just heard about gene expression as a single cell application, ATAC sequencing, immune profiling, and even spatial sequencing as well. So given these areas of interest, each of them does require a different specific readout or a different amount of data to get the information that you need for your experiment there. So we have a variety of instruments in our portfolio here, from some of the smaller benchtops for QC, and then we have some other smaller benchtop instruments for just general lower throughput, and then we have our mid throughput and high throughput. So just to give a little bit of an overview of those instruments here before we move on, I just want to point out that we do have an instrument for almost every application here. If you look to the far end, you're going to see our larger instruments that are going to be having a higher throughput, and you'll see that these are going to be the ones that we're going to be discussing as they are the best suited for high throughput single cell and high throughput spatial genomics here. But on the far end, I do want to note that you can use these lower throughput instruments for doing QC on these runs and balancing libraries before going on to these instruments here. And as you can see here, as the instruments get larger and you can scale up your projects more, the cost per gigabase does decrease. And so with that, we're going to be focusing on the NextSeq 2000 and the NovaSeq 6000. So the NextSeq 2000 is our newest instrument. It bridges the gap very well between our lower throughput benchtop instruments and the NovaSeq. You can, they're a little bit for smaller projects that you might not be able to fill out a NovaSeq flow cell with. And then, of course, the the NovaSeq 6000, which has been out on the market since 2017. It's proven this is an instrument that you can find here at the core facility, and your single cell libraries will be loaded on this as well here. So with that, we're going to be moving into the NextSeq 2000. So with this new instrument that was released, we've had a number of breakthroughs that have allowed us to increase data output, reduce turnaround time, just make the actual workflow much easier to use. We're going to be focusing on 5 of these specific advancements here. The blue-colored 2-lane channel chemistry, just an increase in our optics systems, as well as improving upon our pattern flow cells. We are also going to just talk about briefly the cartridges that we've developed to go along with this NextSeq 2000, which has really streamlined the process. And then also we've touched upon our secondary analysis pipelines, which have really sped up the analysis side of things, as well as reduced data footprint as well by utilizing our Dragon pipelines. So just to go into some background regarding our pattern flow cells. Pattern flow cells, in the past, most of our flow cells were random clustering flow cells where the libraries would flow across the flow cell and hybridize in just random places there. So sometimes you'd get these polyclonal clusters where you might not get a great read from it necessarily. So by developing these pattern flow cells where we have these individual microwells. Only one specific molecule can access that well, and that way actually reduce costs by utilizing our super resolution optics to reduce spending time creating a cluster map and mapping that back and creating that for analysis during the primary analysis side of our pipeline. So by making these smaller and then also utilizing our blue two-channel color chemistry, which has improved upon our current SBS chemistry, which in the past, A, T, G, C each have had their own dye. So by reducing the amount of colors, you're reducing the amount of pictures that you can use or that you need, which reduces the amount of files and storage on your space, so speeds up the pipeline in that way. I'm going to have a picture later showcasing the actual difference in size between the NextSeq 550 and the NextSeq 2000, but by incorporating these advancements here, you are miniaturizing your experiment, taking up less space in your lab. You are also maximizing your data density per flow cell here. So just to show a little bit of a comparison here, and once again, there's going to be an actual picture showcasing the differences in the flow cells here. But you get a 3 times output from that smaller flow cell compared to the NextSeq 550, the previous model there. And then as well as that, because you're miniaturizing and you're utilizing these advancements here, you're getting double the amount, or twice the amount of lower dollar per gigabase for each run compared to the NextSeq 550 with this. So just to give you an idea here, that little box at the very bottom there is the flow cell for the NextSeq 2000. Next to it is the NextSeq 550 there. So through these advancements, we've really been able to miniaturize, and it's just something that we're just constantly trying to improve upon to reduce turnaround time, increase the quality of data that you get, and then also reduce the amount of costs. So just another thing I'd like to note here as well, with this cartridge, the NextSeq 2000 is a completely dry instrument. So all you need to do is take the cartridge, you're spiking in the library, you place it in, and you don't have to worry about any maintenance washes. You just take the cartridge out. We have a kit to take apart the kit, and you can recycle all the components. So we're reducing the size of these instruments. We're just making this as easy and streamlinable as possible for your lab here. And the final thing I want to touch upon is we have integrated our Dragon analysis pipeline onto the NextSeq 2000 to speed up secondary analysis. So our Dragon pipeline, and Dragon also stands for Dynamic Read Analysis of the Genome, so great acronym there, we incorporate a lot of pre-built pipelines there to help out with all of your experimental needs. So we have a built-in pipeline for RNA, we have a built-in pipeline for amplicon, just a variety of gene expression. I do want to point out that we do also have a single-cell pipeline as well that can be utilized when running single-cell experiments on a NextSeq 2000. But even beyond those pipelines, using Dragon to generate FASTQs saves time, and also using our Aura compression also reduces the data footprint that would be necessary for storage. So you're reducing storage costs and increasing time on turnaround. So the final thing I want to note here are the different flow cell configurations, just to give an idea of the levels of flexibility. So with the NextSeq 2000, you are getting access to the P3 flow cell, which isn't available in our NextSeq 1000 model. But as you can see here, and just to kind of give a comparison to our previous NextSeq 550, the P1 is most equatable to our mid-output offerings, or the lower mid-throughput offerings. The P2 is most comparable to our high output on the NextSeq 550. And with the P3, we're starting to get into that range of the NovaSeq there. So this instrument is a great transition for those who might have a little bit of a smaller project, may want a little bit of a faster turnaround time, and you have a different variety of cycle kits here, such as at the end you have a 50 cycle, 100 cycle, 200 cycle, and 300 cycle kit. For the P2, you have the 100 cycle, 200, 300. And then for P1, we just have the 300 cycle kit for some of the smaller projects, if you're just looking for 100 million reads in that sense. So with that, I just want to move on to the NovaSeq 6000 very quickly here. So we have discussed a lot of the advancements that were first found here, or were first utilized here in the NextSeq 6000 and then improved upon in the 2000. So we're not going to go into as much detail on the new advancements in this case, but I would just like to point out that this has been on the market since 2017. A variety of papers online, I wish I had the specific number, have utilized these instruments here to get the data they need for sequencing. And as mentioned, if you go into the core lab, you'd be able to see the NovaSeq there as well. So just to touch upon some of the technical advancements that aren't found in the NextSeq 2000 specifically, one of the main perks is the also added flexibility with the NextSeq 6000. So you can run 2 flow cells at the same time on this instrument here. So it could be, you can be using one of the smaller read throughput instruments and then also running our S4, which is our highest throughput. You can get up to 10 billion reads on. I'll be talking a little bit about the configurations in a second there. But not only that, depending on the flow cell you're using, you can also use specific lanes of each flow cell for your experiment. So say, for example, you want to be using a 10x library, and you also are looking perhaps ATAC-seq libraries. You could split up the pools and load them each into a specific lane using our XP workflows. So as mentioned, we have this will be using a pattern flow cell as well to save time as far as data analysis goes. The NextSeq 2000 has actually improved upon the pattern flow cell and increased the pitch. So just to give an idea that we're constantly thinking about ways to improve upon our workflow and speed up time and get the most data possible for every run that you're running here. So just to touch upon now the NovaSeq system configurations. We have 4 offerings for extra flexibility as well. We have our SP flow cell, which has 2 lanes. And then you can see from SP, S1, S2, 2 lanes. And then we have our S4, which has 4 lanes available. So just to point out, just because the difference in single read output, these are in billions of reads here, as opposed to the NextSeq 2000, which were previously are shown in millions of reads there. So the SP and S1 configurations are for smaller projects, depending on the size, or in this case of 10x, depending on how many cells you might have. This could be an option for you, but as you start to scale up, you can accommodate that based on the S2 and S4 flow cell here. So just to give an example, or just to start to go into 10x now and the library, or how the library might look, and then also how you can start to figure out how many cells or how many reads you might need for every experiment here. So just to talk about the 10x Chromium gene expression library, it looks very similar to a Illumina next generation sequencing library, prep library, and that you have your oligos on either side, the P5 and the P7, which will hybridize the flow cell. You have your primers for initiating the SBS chemistry, and then you also have the indices here. The main difference, though, is for read 1, as mentioned previously, you're actually going to be reading the 10x barcode for that specific sample, and then the UMI for that specific fragment of genetic material that you're sequencing. So the read 2 is actually the insert that you are using to look into or that contains the information in the sequence that you're looking at there. So something to note about this, I had mentioned the 100 cycle kits. Our 100 cycle kits actually provide enough overage so that you can reach and if you have that all up there, 138 cycles there. So you're utilizing every cycle. You don't have to worry about purchasing extra and then not being able to get all the cycles that you paid for there. So starting to talk about some different configurations and some different experiments here. We're going to just focus on the gene expression since the winners will be getting the gene expression library preps here. So you can see with the NextSeq 2000 P2, and the reason we're not showing the P1 is it just doesn't provide enough output for what you were going to be looking for here. When it comes to gene expression, the normal recommendation of reads that you're looking for are 20,000 to 50,000 reads per cell here. So in this case, we have 7 samples with approximately 3,000 cells per sample here. For 20,000 cells, you're going to be getting into that 400 million read range here. And then if we're moving up into the P3 now, you can see that the amount of samples has doubled here in this case here. And 20,000 reads, 3,000 cells, 17 samples. And just to provide some more information too, or just to show a little bit more that when it comes to ATAC sequencing or immune profiling, the considerations and the reads that you need are going to shift. And it changes a little bit more with spatial as well, but we can also load spatial libraries onto these instruments as well. So then just to start to show again, moving into the NovaSeq now, just for the gene expression libraries, we see that we have for the SP flow cell 14 samples here at 3,000 cells, 20,000 reads per cell. And then by just jumping all the way to S4, you can see that we've jumped up to 170 samples, 3,000 cells a sample, 20,000 reads. This is just to give an idea of how you can scale from the NovaSeq and the NextSeq 2000. And so when it comes to designing the experiment too, we have a team here, myself, the 10X team, as well as the core facility here, can help you figure out your experimental needs as you design and make sure that you're utilizing and getting the best instrument and the best configuration that's right for you there. So with that, I actually want to open up for questions now, and thank you all for listening.
So much, Logan. Is there any question?
So Logan.
Yes.
Here at the Biotechnology Center, we only have the NovaSeq 6000 sequencer.
Mm-hmm.
I noticed that you're stating that for the 3' gene expression study, you're only recommending 20,000 reads per cell.
Mm-hmm.
Is that total reads or reads passing filter?
Reads passing filter there, I would say. And in those cases too, depending on what the experiment type is, if you're looking for rare cell types, perhaps you're going to be increasing the amount of reads that we would recommend in that case. So it would come down to experimental design in the long run. And just to repeat the question was the 20,000 read output for pass filter reads or for total reads there? And just for the people at home right now.
Thanks.
Thank you.
Thank you. I'll introduce you, then we can turn it on. Is that okay? It's off right now. Sure. Well, thank you. I just wanted to introduce our next presenter. So Dr. Saha is actually Associate Professor at the University of Wisconsin-Madison, presenting today in a single-cell technology seminar. So thank you so much.
It's really a pleasure to be here, and thank you for the invitation. Certainly, our work is enabled by all of the great support we get from the Biotech Center as well as from 10X. And today, I think I'll give you more of a conceptual talk and two examples of where we've used single-cell RNA-seq. The other strategies that we've been using have been single-cell attack-seq as well as single-nuclei RNA-seq. But today, I'll focus on the I guess, a bit older single-cell RNA-seq. Our work here is really to develop new human therapeutics, specifically both gene therapies and cell therapies. And we use a variety of platforms, including human pluripotent stem cells that could be patient-specific. That's what's shown here is a retinal organoid that we've treated with genome editors. And we can monitor this in real time using some metabolic imaging techniques from our collaborator, Melissa Scala. That's what's shining, I think, if this video plays. In the middle with these green outer segments that are responsive to light. So we really get nice functional measurements out of this. But as you can tell, these are very complex types of culture systems, and we do want to have single-cell resolution to understand both what the effects are of particular gene therapies in that case. So just to remind folks, when I talk about genome editing, these are changes to the DNA code when we do DNA-based editing. And this example is a mutant allele in BEST1. This T allele could be corrected or edited more specifically by using CRISPR-Cas9 technologies as well as other genome editing technologies to the proper base that would, in theory, rescue this type of visual defect in central vision, as this depiction of their visual defect in central vision is shown here. And so I'll tell you today one story on this BEST disease gene editing strategy that we've used 10x technology to look at potential adverse effects from genome editing. We are also doing work in a number of other retinal disorders with collaborators here on Pompe disease, a autosomal recessive condition, a number of other disorders also affecting the brain. And then the second story will be on actual ex vivo gene-edited cells, namely T-cell therapy for cancer that we've profiled using 10x technology as well. And in general, we have a number of projects here that take advantage of a federal consortium that was funded about 4 years ago, and phase 2 is just about to get launched. It has initiatives that are trying to make a modular toolkit to advance therapeutic in vivo gene editing. So there's specifically new delivery systems. There's also new animal systems to test those editors, new types of editors themselves that are being shown in this video. And in general, there's a range of editors that could be theoretically packaged into these delivery systems and put into the body to edit specific cell types in particular tissues. Today's talk is really on Cas9, but as you can see here, there's epigenetic editors, there's base editors, there's prime editors that we're also working on. And so, today's talk is going to put a plug in for human cell-based platforms, specifically stem cell-based platforms that Wisconsin is known for pioneering. And one of the highlights here with single cell profiling is to look at transcriptional perturbations upon delivery of the editor. And so in this case, what we've looked at is, again, BEST disease that I mentioned. And normally, you would administer the gene therapy through subretinal injection, that purple bleb in the back of the eye, to hit the pigmented epithelial cell layer, the RPE, that is where BEST1 is expressed. The challenges here, and one of the reasons why we chose to go with human stem cell-based models, is that there is no good animal model that mimics what happens in patients, and therefore, really the best option is to use a human cell-based model. And really the relevant target is in the human genome, not in a mouse genome or in a dog genome. And we've taken advantage of the retinal organoid and as well as directed differentiation towards pigmented epithelial cells that's shown here through our collaboration in the GAM lab. And through that collaboration, we've had access to patient-specific induced pluripotent stem cell lines, namely from 4 patients that have different mutations in BEST1. And we tested a variety of gene therapy techniques, traditional gene augmentation that's shown on the top right, as well as mutant allele-specific editing to correct that error using CRISPR. And I won't go into all the details of how the editing works, but rather highlight the single-cell profiling part of this story. Both of these strategies do end up providing functional rescue of this phenotype that you can see in terms of chloride and chloride ion channel dysfunction in these RPE layers. But we wanted to see whether the expression of the editor itself would have a problem. And so our promoter here in our lentiviral delivery construct is driven by the endogenous BES1 promoter. And so what we did was use those patient-specific iPS cells, dissociated them, and then brought them over here to the biotech center, loaded them onto the Chromium system, and did single-cell RNA-seq. You can see 12,000 cells here in a T-SNE plot for 2 genes, BEST1, that's expressed endogenously, and in that case, it would be the mutant or the corrected form. We can't distinguish that with this 3' capture. And then also our transgene Cas9 GFP, which we can see in each of these cells. And the expression level between, comparing those 2 genes in these cells is identical, which is good, which means that our expression of our editor is indeed driven by the BES1 promoter. And if we look at pairs of samples now between delivering a control guide RNA, which would end up cutting not at the BES1 locus, just in a control region of the genome, and then one where we're cutting at a specific mutation in BES1. There's 3 different examples here, A14K, amino acid change, N296H, R218C. And we compare what we see in our T-SNE plots. Between the two, you essentially see almost identical type of clustering, meaning there's no overt changes in cell types that we see in these edited cell cultures. And then on the far right, what we've done is correct the mutation at the stem cell stage and then gone through the entire differentiation and application of gene therapy. And there, as expected, if you look at the differential genes on the second middle type of row here, there's almost no change between the left and the right, except for this one sample that came up in our control that had a number of genes differentially regulated that related to DNA damage response, apoptosis, cell cycle, and an innate immune response. That's consistent with a few other studies that have looked at RNA-seq of edited hematopoietic stem cells where they see a response to the viral editor. And so this allows us to make a conclusion here that actually our gene editing strategy itself is not necessarily the cause of significant perturbation because this was in our control sample, but there may be some issues related to delivery that we should think about. We are also looking at the other tissue that would be seeing the genome editor in a patient, which would be the photoreceptor or neural retina layer. And there we have to switch to another set of culture techniques to make the photoreceptors, because they don't attach naturally, at least in vitro, to the retinal pigment epithelial layer. And so this is a longer-term project supported through that NIH consortium where we've added our genome editors to this organoid and then done single-cell RNA-seq, as well as some of the metabolic imaging that I showed at the first slide. And the hope here is that this type of workflow or pipeline would help us identify any transcriptional perturbations that might be related to genotoxicity, perhaps immunogenicity, or any other type of adverse event on the visual cycle within these cells. And so we're midway through this. I'll report some interim results where we've done this now for, I think, on the order of I believe around 80,000 cells. This is just the first 2,000 cells that I'm showing here. We do get the expected distribution of cell types. It does depend on how long we differentiate these organoids in culture, and certainly, so that's one area of investigation. But we can identify the ones that receive the genome editor. In this case, we had fairly low delivery, about 13%. There are ways that we are working on to increase this to 60 or 70%, but it does require some dissociation prior to delivery. But even with this type of smaller data set, we've done some analysis, and this is primarily through Sushmita Roy's lab here across the street at WID, who's looking at the gene regulatory networks within these cells that have the editor, that are called editor plus cells, and those that do not. And then here the intensity of red is the amount of network overlap in these gene regulatory networks. And what we see is that there's more intense red between the cells that have the editor than those that don't, and vice versa. Those that don't have the editor are more similar to one another than those that do. And so there is a signature here of transduction that we are looking at, and also we're looking more deeply at whether this relates to the targeting of the editor or not. So that's the first story on gene editing therapies and how we've used single cell technology. The second story here is on ex vivo manufacturing of cell therapies, and this is a different center that we've been working with, and that's the NSF Center for Cell Manufacturing Technologies. And in general, this center has a number of test beds. We've been focused primarily on the T-cell test bed, and the idea is that you would be able to eventually engineer a cross-cutting system, essentially a black box that has feedback control, such that if you detect changes in your product in line, you could come back and change process conditions to get it back on specifications. And so we're applying a number of techniques, imaging, big data analysis, and some fundamental knowledge from biology. And my lab is focused primarily on the gene transfer step and gene editing. And so here, the product is not direct injection into the eye, like Luxturna in the top left, but rather infusion of a cell product. And in this case, CAR T cells have 5 to 6 approved products already now being implemented in the clinic. All of them to date use viral vectors, and for introducing the functionality, which is the chimeric antigen receptor. And we've been using CRISPR instead, without any viruses, to insert the chimeric antigen receptor. And so what we're doing is cutting into the T-cell receptor alpha locus or track locus, and knocking in a CAR, which is this multicolored sequence in the donor template. And this is a target for anti-GD2, a tumor-associated antigen on solid tumors. And one of the questions we had is, how does this compare to standard techniques used in industry? And so this last bullet point, the memory-like phenotypes that we've found really were enabled by using single-cell RNA-seq, which I'll show you in the next slides. So our comparison have been head-to-head of our virus-free CRISPR cells in blue in the middle versus a traditional retroviral delivered cell on the left that would mimic that you would see in approved products. And then what we've also done is engineered a virus-free control cell on the right that does not have the chimeric antigen receptor, but rather has an inert mCherry transgene. And you can see our gene transfer efficiencies are relatively similar. If you look at the chimeric antigen receptor on the y-axis in the flow cytometry plot, it's about 50% inserted. And then on the x-axis, we're looking at TCR, T-cell receptor, and in our CRISPR versions, we're knocking that out, and therefore, in those cases, we have 90% plus knockout as well. So does the knockout matter? Does the CRISPR matter? And so we profiled here about, again, 80,000 cells through the Biotech Center. And this is a T-SNE for all of those cells. And we clustered them using Seurat and found approximately 17 or so clusters that we could identify with known markers of memory and effector. and partial exhaustion states of T cells. And the way that we've colored this plot right now is to highlight the memory-like phenotypes in purple, and intensity of purple, it means would more known early stem-like memory marker genes, and then the more intense yellow is the more effector and partially exhausted like clusters. So if we have a violin plot of what these marker genes are, here are just 5 examples of that. It gives us a good sense of where memory and exhaustion lie, and it looks like it's kind of a diagonal gradient from top left to bottom right of going from exhaustion and memory. And so there's a lot of detail here. We have CD4 and CD8 cells and a spectrum of cell types, and this is both during manufacturing, and then we've also looked at the response after co-culture with the cancer cell that have the CAR target. And the top here in C is what you would get at the end of manufacturing prior to infusion. And at the bottom is what you would see after co-culture with a cancer cell, kind of a potency assay that you would use for releasing a batch, for instance. And on the x-axis here is the percentage of cells in a sample within each cluster. And what we notice here is that the blue VFC CAR ones have a longer purple bar relative to a donor-matched exact same CAR delivered using viral techniques, the RV green bar. And that's the case in the pre-antigen or case as well, a bit in the post-antigen, although both of them are responding to the antigen quite robustly. So you certainly see the shift to more effector-like cells in yellow after you co-culture, which is expected. And so this gives us a sense that really before putting these cells into the animal models in our case, or potentially patients, that these cells would have a more memory-like phenotype. And in our case, I think that's correlated to better responses in the clinic, as many others have observed through their clinical trials to date. And so we are working with the UW program for Advanced Cell Therapy, or PACT, out in the hospital. They have a GMP manufacturing space, and there's a clinical collaborator here as well, Christian Capitini, that has worked on us from the very beginning on this type of strategy. And so this profiling has enabled us to look a bit more deeply at the infusion states, both during manufacturing with respect to memory, and also led to some hypotheses of what could happen in vivo relating to differentiation, memory, and exhaustion. And so I'll stop there to give a few minutes for questions. In summary, I think I've given you 2 stories that there's some references here if you're interested for more detail on how we can probe both potential adverse effects from genome editing, as well as some of the functional outcomes, as well as differentiation states of the cells after gene editing as well. So with that, I'll be happy to take some questions.
Looking ahead, what do you kind of foresee over the next year, knowing the capabilities you have here with the Biotech Center, and where this is going? What does it look like for the next year?
Yeah, I mean, we have some active projects to see exactly what's happening in vivo to those T cells after infusion. There's mice right now with T cells in their spleen. I believe that we'd like to really understand when are they differentiating, why are they differentiating, what's happening in response to various microenvironmental cues, maybe to aging. We have other CAR T-cell products that are not specific to cancer, but are interested in neurodegeneration and aging that we'd like to see what's happening in the brain. And then we are also injecting these CRISPR strategies into the brain as well. And so we have a project there to understand which cell types are we hitting, are they still functional? So those are some areas that single cell profiling are really hypothesis generating. You know, I'd love to propose that we would do quality control using single cell, but most of the response out there has been that it's overkill, and potentially too expensive. So using this type of profiling to maybe find some biomarkers that could be done in a reduced panel, certainly there's plenty of interest there. I'd love to be able to do the whole thing, but I know that's challenging to do, but perhaps 10x has a solution. One of the other, I think, exciting areas in the field is the idea of doing off-the-shelf allogeneic products. And there's a great need to be able to do multiple rounds of gene editing at the stem cell stage. So putting in the chimeric antigen receptor, cutting out checkpoints, and doing this really, you know, up to 5, 10, even 20 edits I've seen proposed. And you'd like to get a kind of workflow or a pipeline to do that quickly and efficiently. And there's certainly opportunities to profile those outcomes you know, more thoroughly, because once you cut the genome, there's a whole spectrum of different outcomes, and being able to tie this transcriptional readout to a genotype is something that I've seen done out in a few papers, but you know, I haven't seen done with respect to CRISPR in particular yet.
Thank you so much, Dr. Sarah. Really appreciate your presentation today. Thank you.
Thanks for having me.
Thank you. Well, thank you everyone for attending virtually and in person. So without further ado, I wanted to actually introduce the CoreGram Lab program, which will actually provide 2 people at the University of Wisconsin-Madison to get 2 samples actually from 10x and sequenced by Illumina. So I'll describe the program. So it will be submitting a maximum of 300 words abstract. And the link is on actually the Genomic Core Facility website, so we'll be able to get it out as well, how to apply. So the idea is to have a pilot project that is tied to a grant submission. So we wanted to help 2 different labs to get pilot data to prove show of concept with single cell with a 10x genomic assay. So either the gene expression 3' chemistry or the 5' immunoprofiling chemistry. And we will have actually a committee that will pick 2 winners, as I said. The criteria will be on research, creativity, and innovation for a research project. And we will be taking application from today, March 22nd, until April 22nd, so 4 full weeks. And the 2 samples for the winner have to be submitted to the Genomic Core by June 3rd. So again, we'll be picking 2 winners, providing 2 samples to be profiled with the 10x genomic gene expression kit. So thank you so much, and I look forward to read all the application and to see the creativity going on campus. And so I would like to thank all the speakers today and the Genomic Core Facility for all their support, Illumina as well. So thank you so much, everyone. And yeah, we can take questions definitely after. You can also submit your question via the chat, and we'll be monitoring those as well. So thank you again, everybody. It was a pleasure to be here.